The human brain, a marvel of evolution, a complex, interconnected universe within our skulls, capable of orchestrating symphonies, solving equations, and dreaming of distant stars. Yet, this same magnificent organ, designed for survival and flourishing, harbors a profound vulnerability. A susceptibility to hijacking, to manipulation, to the insidious whispers of addiction. Imagine a single, unassuming cell. A neuron. It fires a tiny electrical impulse, sending a message across a synaptic gap, a chemical bridge to another neuron. Billions of these interactions, every second of every day, construct our reality. They dictate our thoughts, our feelings, our very perception of self. Now, picture that delicate balance disrupted. Imagine a foreign entity, a molecule, a substance, or even an extreme behavior, crashing into this intricate network, not merely altering a signal, but rewriting the very code of desire. This isn't a hypothetical horror story. This is the reality for millions. For the individual who cannot stop gambling, even as their life crumbles. For the one consumed by an insatiable craving for alcohol, despite all consequences. For the person trapped in a cycle of digital obsession, sacrificing sleep, relationships, and well-being. What force possesses them? What architect designs these invisible chains? The answer, disturbingly, lies within their own minds. It is the brain, in its relentless pursuit of pleasure and avoidance of pain, that becomes the unwitting forge of its own undoing. It is a story of neurochemistry, of evolutionary design gone awry, and of the profound, often terrifying, power the brain wields over its host. We are, in essence, our brains. And understanding how this intricate biological machine can be reprogrammed, how it can betray its own best interests, is not just a scientific endeavor. It is a journey into the heart of human struggle, a quest to comprehend the invisible forces that shape our choices, our freedoms, and ultimately, our destinies. This is the story of how your brain creates addictions, a narrative woven from the very fabric of your biological being. At the core of this vulnerability lies the brain's most ancient and powerful motivational system, the reward pathway. This isn't just a simple on-off switch for pleasure. It's a sophisticated network, honed over millions of years of evolution, designed to ensure our survival by reinforcing behaviors essential for propagation: eating, drinking, mating, and social connection. When we engage in these life-sustaining activities, a cascade of neurochemical events unfolds, culminating in the release of dopamine, often dubbed the brain's pleasure chemical. However, this label is a significant oversimplification, one that obscures dopamine's true, more insidious role. Early research by Olds and Milner in 1954 provided a glimpse into this system's astonishing power. They discovered that rats would incessantly press a lever to receive electrical stimulation in a specific brain region, later identified as part of the mesolimbic dopamine pathway. The rats would forego food, water, and even mating to continue stimulating this area, illustrating a drive so potent it superseded basic survival instincts. This wasn't about liking the sensation, it was about wanting it, an intense motivational pull that researchers like Kent Berridge and Terry Robinson later meticulously differentiated in the 1990s and early 2000s. They proposed that dopamine primarily mediates wanting, or incentive salience, the motivational drive to pursue a reward, rather than the actual liking or hedonic experience of it. This distinction is crucial. Addictive substances and behaviors don't just make us feel good, they profoundly amplify the desire for that good feeling, turning a whisper of interest into a screaming imperative. Consider the journey of dopamine. It originates in the ventral tegmental area, or VTA, a cluster of neurons deep within the midbrain. From there, dopamine neurons project to several key regions, forming the core of the reward circuit. One primary destination is the nucleus accumbens, a critical hub in the brain's pleasure and motivation center. Another is the prefrontal cortex, the seat of executive functions, decision-making, and impulse control. When a natural reward, like a delicious meal after a long hunger, is encountered, the VTA releases dopamine into the nucleus accumbens. This surge signals to the brain, "This is important. Do this again." It encodes the experience with a powerful motivational tag, strengthening the neural pathways associated with that behavior. Now, introduce an addictive substance, such as cocaine. Unlike natural rewards, which elicit a controlled, transient dopamine release, cocaine directly and dramatically blocks the reuptake of dopamine in the synapse. This means dopamine floods the nucleus accumbens and lingers there, generating an intense, prolonged, and unnatural high. The brain is overwhelmed. It registers this experience as exponentially more rewarding than any natural stimulus. The result is an immediate, powerful, and often unforgettable surge of wanting, an almost irresistible urge to repeat the experience. The same principles apply to highly addictive behaviors. Gambling, for instance, triggers dopamine release not just with a win, but with the anticipation of a win, especially under conditions of intermittent reinforcement where rewards are unpredictable. This variability, this tantalizing uncertainty, keeps the dopamine system in a state of high alert, constantly driving the wanting for the next potential payout. The brain becomes exquisitely tuned to the cues associated with the addiction, transforming benign environmental triggers into potent signals that ignite desire. A specific street corner, the sight of a casino, even the sound of an online notification. Each can become a powerful reminder, activating the dopamine system and initiating the compulsive pursuit. This brings us to the second critical mechanism, neuroadaptation and the insidious creep of tolerance. The brain is a master of homeostasis, always striving for balance. When it is repeatedly bombarded by unnaturally high levels of dopamine or other neurotransmitters due to chronic exposure to drugs or intense behaviors, it attempts to restore equilibrium. It does this by making adaptations at the cellular level, essentially turning down the volume on its own reward system. One primary way the brain adapts is through a process called downregulation. It reduces the number of dopamine receptors on the surface of neurons in the nucleus accumbens and other reward-related areas. Imagine your brain's listening posts for dopamine. With constant, overwhelming stimulation, it starts to dismantle some of these posts, or make them less sensitive, to protect itself from overstimulation. The consequence? The same amount of the substance or behavior no longer produces the same effect. This phenomenon is known as tolerance. A person who once achieved a profound euphoria from a single dose of heroin now needs 2, then 3, then increasingly larger amounts just to feel normal, let alone high. The brain, having been saturated, literally becomes less responsive. Consider the chronic coffee drinker. Initially, a single cup provides a noticeable jolt of alertness and focus. Over time, that single cup barely registers. They need 2, 3, or more to achieve the same feeling. While caffeine addiction is typically less severe than, say, opioid addiction, it illustrates the same fundamental principle of neuroadaptation. The brain has adapted to the constant presence of caffeine, reducing its sensitivity to the stimulant. The natural, intrinsic rewards of life also become dulled. As the brain downregulates its dopamine receptors, the pleasure derived from eating a favorite meal, enjoying time with loved ones, or engaging in hobbies diminishes. These activities, once sources of genuine joy, now feel bland and uninteresting. The addicted brain, in its desperate state of adaptation, requires the intense, artificial stimulation of the drug or behavior just to feel anything approaching normal. This creates a vicious cycle. The drug is needed more and more to achieve less and less, pushing the individual deeper into the spiral of dependence. The joy is fleeting, the baseline shifts, and the pursuit becomes less about pleasure and more about avoiding the profound discomfort of its absence. Beyond the initial rush and the subsequent adaptation, addiction is profoundly shaped by the brain's stress response and the formation of powerful, often debilitating, cravings. This intricate interplay involves multiple brain regions, most notably the amygdala, the prefrontal cortex, and a network of stress-related pathways. George Koob and Michel Lamole, in their influential allostatic model of addiction from 1997, described addiction as a process where the brain's attempts to maintain stability, homeostasis, in the face of chronic drug exposure lead to a new, pathological setpoint, allostasis, characterized by negative emotional states and an overwhelming drive to seek the drug. The amygdala, a small almond-shaped structure deep within the temporal lobe, is the brain's alarm bell. It processes emotions, particularly fear and anxiety. In the context of addiction, the amygdala becomes hyperactive during withdrawal, contributing to the intense dysphoria, irritability, and anxiety that plague individuals trying to quit. This negative emotional state isn't just unpleasant, it's a powerful driver of relapse. The brain learns to associate the drug or behavior not just with pleasure, but with the alleviation of withdrawal symptoms and negative emotions. This shift from positive reinforcement, seeking pleasure, to negative reinforcement, avoiding pain, is a hallmark of the transition from recreational use to compulsive addiction. The addict isn't necessarily chasing a high anymore, they're desperately trying to escape the profound low. Cravings themselves are complex phenomena, more than just a fleeting desire. They are powerful, often intrusive thoughts and urges that can hijack attention and decision-making. These cravings are heavily influenced by classical conditioning. Just as Pavlov's dogs learned to salivate at the sound of a bell associated with food, individuals with addiction learn to associate specific cues, people, places, objects, moods, with the drug experience. For someone addicted to alcohol, the sight of a bar, the clinking of ice in a glass, or even a stressful argument can become a potent trigger. These cues activate the same reward pathways that were initially stimulated by the drug itself, unleashing a torrent of dopamine and igniting an almost irresistible craving. This is why individuals in recovery are often advised to avoid triggers. These aren't just psychological suggestions. They are attempts to prevent the brain from reactivating deeply ingrained neural associations that can overwhelm even the strongest resolve. The prefrontal cortex, the brain's executive control center, plays a crucial role in regulating these cravings. It's responsible for planning, decision-making, impulse control, and assessing consequences. However, chronic drug use or compulsive behaviors significantly impair the function of the prefrontal cortex. Neuroimaging studies have shown reduced activity in the prefrontal cortex of individuals with addiction, particularly in areas associated with inhibitory control and rational decision-making. This means that the very part of the brain designed to say, no, is weakened, making it incredibly difficult to resist the powerful, yes, emanating from the hyperactive reward system and the fear-driven amygdala. The capacity for long-term planning, for weighing the severe consequences of relapse against the immediate relief offered by the drug, is severely compromised. The addicted brain is, in a very real sense, operating with a diminished capacity for self-governance, caught in a relentless struggle between an overactive, wanting system and an underactive, stopping system. The individual knows the choice is destructive. They know the path leads to further suffering. Yet the brain's own machinery has been rewired to prioritize the immediate, albeit fleeting, escape over long-term well-being. The journey into addiction often begins with conscious choices, but it rapidly morphs into something far more automatic and ingrained: habit. This transition from volitional action to automatic routine is mediated by the basal ganglia, a group of structures deep within the brain, particularly the dorsal striatum. This system is responsible for procedural learning and the formation of habits, both good and bad. When we repeat a behavior consistently, especially in the same context and with a consistent reward, the brain begins to streamline the process. The initial conscious effort required to perform the action diminishes, and it eventually becomes an automatic sequence, a habit loop. Charles Duhigg, in his work on habit formation, describes this as a cue routine reward cycle. For addiction, the cue might be stress, boredom, or a specific environmental trigger. The routine is the addictive behavior itself, and the reward is the chemical surge or the alleviation of withdrawal. Over time, the link between the cue and the routine becomes so strong that the behavior is initiated without conscious thought, almost reflexively. Consider the individual who smokes. Initially, they might consciously choose to light a cigarette in social situations or to relieve stress. Each time they perform this action, the neural pathways linking the cue, stress, social gathering, to the routine, lighting up, and the reward, nicotine's effect, are strengthened. Eventually, the very act of finishing a meal, or stepping outside, or experiencing a moment of anxiety can automatically trigger the urge to smoke, regardless of whether they truly want a cigarette. The behavior becomes deeply embedded in the dorsal striatum, bypassing the more reflective, effortful processing of the prefrontal cortex. Studies using functional MRI have shown increased activity in the dorsal striatum in individuals performing habitual tasks, even when those tasks are no longer rewarding. This suggests that the habit system operates somewhat independently of the reward system, driving behaviors even in the absence of pleasure. This habitual component is particularly insidious because it underlies the compulsive nature of addiction. Even if the immediate pleasure from a drug or behavior has significantly diminished due to tolerance, the learned habit of engaging in it persists. The brain has created a powerful shortcut. It's no longer a matter of choosing. It's a deeply grooved pathway that the brain automatically follows when confronted with the right cues. This is why relapse can occur even after long periods of sobriety. A familiar sight, a specific smell, an unexpected emotional challenge. Any of these can activate the ingrained habit loop, bypassing conscious intent and initiating a powerful, almost overwhelming, drive to engage in the addictive behavior. The brain is literally running on autopilot, executing a program that has been deeply etched into its neural architecture, making the fight against addiction a battle not just of will, but against the very mechanics of learned behavior. The individual might intellectually understand the harm, they might deeply desire to stop, but the brain's ingrained habits exert a relentless, often subconscious, pressure, making the path to recovery an arduous and constant struggle against their own neurological programming. The final, and perhaps most devastating, transformation wrought by addiction is the shift from impulsivity to compulsivity. These two concepts, while seemingly similar, represent distinct neurological processes and drive different facets of addictive behavior. Impulsivity is characterized by acting on a sudden urge without much forethought, often driven by the immediate pursuit